Okay, well, I'm delighted to chat with you today about. Now, we didn't actually title the talk, so we'll see how deep it is. All right, hopefully it's as deep as anticipated. Deep text mining for cancer disease. I'm going to be spending less time, getting all the way to cancer and disease, and more on the text mining and what different processes and steps in that mining can reveal about the game of science in general. So this is James Cameron, who, with his wealth, following two enormously blockbuster films, decided that he wanted to explore the ocean floor, right? And so he kind of dives down to the Mariana Trench and looks for strange creatures. Our claim here is that human knowledge ends up getting really buried in this deep trench of published literature, and this graph here is basically traces out kind of a log normal distribution. If you look across journals and across papers, log normal distribution of kind of attention decay over time, right? So the half-life of a paper across all the journals in science scholarship is about 4.5 years. If you just look inside cancer biology, you can kind of scale that back to about 3 years. So you have this enormous attention deficit, if you will, like this, a huge mountain, or in this case, trench full of papers that really get very little attention. It's too big to manually read. It's too diverse to really manually assemble. And what I mean by that is there's so many different scientific languages, right, or sub-languages which are involved across these scientific cultures that you can't really align the nouns and the verbs trivially, right? They don't align themselves. And so digital documents end up being this large repository, and the question is, how do we extract information about these claims themselves and the game of which they are part, so that we can kind of actually feed that back into our certainty of those claims, and then feed them into a broader analysis of how we can combine that claims with experimental high throughput data, with medical records, with other kinds of information. So how does one extract knowledge from papers? Well, there's the standard pipeline, right? And we're using this standard pipeline, as well as what I'll talk about in a minute, is kind of a non-standard pipeline. So the standard pipeline is that you extract a set of concepts which involve both nouns, right, or arguments. So these nouns are things like genes, proteins, drugs, diseases, methods, and verbs, right, which end up being used as predicates, right? So phosphorylate, methylate, upregulate, etc., based on very domain-specific terminologies, ontologies, and you extract these from some given corpus of documents. So that's stage one, right? It's extracting these elements which are kind of defined to be members of a certain class of elements. The second is that you normalize those concepts, right? So you engage in some kind of disambiguation. That disambiguation can be complex, right? It could be that these terms have many-to-many relationships between the strings that you're observing and the meanings. It could be that they have one-to-many or many-to-one. So you want to, for example, unify synonyms, things that mean the same thing. You want to kind of pull them together. Homonyms you want to split, so things that you say describe them in the same way and yet they mean multiple things. You want to kind of separate them into their semantically identifiable units. And then finally you assemble these into statements, right? So you parse the arguments into the predicates, right? So you take those nouns effectively and you make them arguments of verbs in a variety of different ways. The most common are these triplets: subject, verb, object. This does something to that. But you also have intransitive verbs, right? Some subjects do things, not to anything else. And you also have kind of verb object. You have processes which end up linking to other things. These can also be recursive, which is to say a subject, verb, object triplet can itself be a subject or an object, typically in the way in which these things assemble over time. So this is the standard pipeline. How else do we extract knowledge? Because knowledge is not simply about the claims that are made, but the claims that are made relative to this population of broader claims, right? And so there's a broader kind of contextual pipeline that we also apply to extraction of information. So you want to embed these concepts and these assembled higher-level statements into contexts. So what context do we care about? Well, a whole variety of contexts, right? So there are framing contexts, right? So there's like citations, there's literature that you bring to bear which condition the classes of questions that you're going to ask, right? That ends up shaping the way in which you understand what's subsequently stated or claimed. There are also a series of methods, right? There are procedures, there are algorithms, there are tools. That provides another kind of context in which these statements arise. So it may be that you're just parroting or amplifying a statement from prior literature. It may be that based on some method you have derived a finding, right, which is this new claim, right? You're inferring this claim. And then that context, the context of, the relative accuracy, the relative importance, the relative frequency of the procedure ends up kind of conditioning and shaping the way in which we think about that statement. Similarly, authors and author teams end up constituting an essential context. Host institutions, the University of Chicago, Harvard University, UCLA, and funders, right? DARPA versus, the National Science Foundation end up kind of conditioning the context of certain kinds of statements. I know, at least for me, for example, the one of the projects that's funding a joint project with Ishanu and Andrei Brzezinski and myself is DARPA, and their approach to this project is much more unusual, say, than the standard National Science Foundation grant that I get. Another context is just the time and place, and by place I mean broader national context in which a statement arises. Or it could be conditioned on a whole host of other things, exogenous information in the world. So in particular with the context of health, the context of a claim becomes more or less relevant. It means something different whether or not that health problem is prevalent or not prevalent, whether it's serious or less serious, right? Whether it occurs in the context in the society that you're analyzing at more or less kind of disability-adjusted life years, whether it costs more life, basically. And the degree to which it ends up being influenced by markets or the anticipated markets for a therapeutic to deal with that medical condition or the popular imagination. And then finally, what do you do with these contexts? Well, you can use them to feed back an ability and the importance of a statement as a function of different aspects of these contexts. So, for example, the attributions in the document itself, right? If someone expresses uncertainty, right? If they say, well, this may be the case, right? So these modifiers end up kind of ornamenting a statement. You can directly end up scaling a statement as a function of those attributions, but these contexts also themselves shape them. So, for example, I'll talk a little bit later, you might, for example, attribute greater strength to a claim if it's repeated frequently in the literature. You might accord it greater strength if it attends or draws to it more citations in subsequent work. You might give it greater strength if it has more independent support, which is to say that socially disconnected groups are promoting or claim the statement, right? So that you don't have these communication-induced dependencies where you can just recruit other people and other problems to the same statement. If you get 4 or 5 or more independent groups which are all making the same claim, then that has you're drawing more balls from the kind of urn of truth. Again, that's not visible from the statement itself, but only from these contexts. You can also assess the relative accuracy, for example, of an individual team or author or a method relative to the population of those authors against a claim. So, for example, if an author is always in the tail of the distribution, relative to the mode of an assessment about a particular claim, about, for example, a protein in its relationship to some deleterious pathological outcome, then you might accord it less strength overall than one that always ends up predicting the mode of the eventual distribution of that claim. And finally, kind of disentangled visibility, which I'll talk about a little bit later. But the idea is, we bring these contexts in to then reshape our certainty in the way in which we think about these claims. And my argument today is that each of these steps which is to say, the extraction of concepts, the normalization concepts, their assemblage into statements, and these various embeddings ends up yielding clues about the relative strength of claims, the distribution of those claims, and about the game of science of which they are a part. And so I'm going to talk a little bit about a handful of these different clues that we kind of get along the way and what we can learn about these clues. So the first is just, the very first step is we extract the concepts from the literature. What can we do with those? So in this case, we take those as just, a distribution of attention to, in this case, various diseases. So we're kind of interested in how is it that the system attends to various diseases over time, and what does that say about how the system thinks about those diseases. Part of this is because we're looking at this not only in the context of the literature, but in the context of the grant distribution, the context of commitments to clinical trials, and in the context of kind of actual insurance claims and a kind of disability-adjusted life year survey, right? How much does each disease cost society? And so we're trying to look at how it is that the concept distribution responds to either the private literature or the prior system of grants or the needs of society. And we find when we look at it at the global level that there's no correlation. In fact, a slight negative correlation between the distribution of research articles and the disability-adjusted life years that are lost as a function of disease, right? So there's no relationship between the kind of the burden or the need and the research investments. And it turns out, if you look at these across countries, so these are just a different class of disease, although we've split them out also into about 120 different specific diseases. You see that, part of this is accounted for by the fact that if you look at the relative GDP, right, of each country, that, different diseases, or I'm sorry, different countries have different distributions of kind of disease burden, right? So poor countries are much more likely to have infectious parasitic disorders, cause more pain and suffering in their societies. Rich societies are more likely to have cancers, malignant neoplasms, and musculoskeletal diseases, arthritis, right? So which are effectively old people's diseases, right? Because they're so successful at kind of pushing people to that part of the distribution. So this is the relative difference in those distributions, right? And you can see, kind of Southeast Asia and Africa and, Central America end up having a very different disease statistical profile in terms of its needs than the developed world. Similarly, kind of linked to that, you've got the number of biomedical articles end up being, related linearly, really, to gross domestic product, right, per capita. So basically, rich countries are producing more articles, which are more relevant to their distribution of diseases and needs, and this ends up accounting for the vast majority of the global disconnect between research investments and actual problems. But when we look at the U.S., and we actually become much more specific than looking just at, for example, the disability-adjusted life years on a few diseases. We actually go into medical insurance claims and look at the full complement of ICD-9 codes, which trace about 15,000 diseases, and all of the insurance claims made against those diseases for about a third of the U.S. population. We find even just within the U.S. and linked to just U.S. publications that there's virtually no relationship or correlation. So this is kind of a 3D correlation plot. There's virtually no correlation between burden and publication. And in fact, for certain populations, so it turns out there's no correlation for those who are over 65. It's slightly negative for those who are 65 and under. And so this is just the relative distribution of basically the relative ranks of age and disease. So you can see there's a little bit of a difference in those who have older diseases and those who have diseases associated with younger people, and that the older diseases kind of attract more relative attention than the younger diseases in the U.S. And so from these kinds of extractions, we can create things like, in this case, a health research opportunity index, a health ROI, which can kind of identify where there's a research opportunity for investment, right? And so the idea is you normalize each of these variables, and then you look at the relative distribution of these variables laid on top of one another. So, for example, this is the full distribution of opportunity, and you can see the opportunity index for breast cancer, for example, is very low, and you can see why. It's not just because there are many publications, although there are. It's driven by the disproportionate nature, or the disproportionate number of clinical trials, which are kind of in place, which represent large capital-intensive commitments to breast cancer. So this can kind of bring out those diseases which are kind of overstudied relative to the burden in society, those diseases that are understudied relative to those in society. And we can also look at those changes over time, whether they're monotonic, whether in some cases they exhibit spike and cyclical dynamics in that attention space. Now, of course, there are many reasons why it is that we might study diseases that are linked or correlated with previous publications and not with needs. And it may be that we actually know how to do something with those researches that have kind of been performed before. So if research is not linked to the disease distribution, or I should say it does look like the overall score is increasing, which is to say that there is this is an imbalance index, that basically there's less imbalance over time across the years in our sample, right? So you have from 2000 to 2011, those distributions end up becoming slightly more aligned, but there's a long way for them to go. This is it would take decades and decades, about 3 decades for them to kind of be approximately aligned. And it turns out that almost all this action takes place at one bump right here between 2004 and 2006, when effectively they stabilize again. So it's not like it's progressively moving towards a better match. So if they don't, if these attention disease doesn't end up corresponding to the needs of society, then what does it correspond to? And for that, we kind of engaged in a study where we looked at the combinations of these concepts across papers, across the PubMed and Medline database. So about 20 million articles, about 10 million authors. And so we took an article like this, NAP protects hippocampal neurons against multiple toxins, and we extracted all of a variety of different named entity elements, right, including disease and phenotype, chemicals of various types, methods, as also things like authors, institutions, etc. And we kind of conceived of each paper as being a hyperedge and a hypergraph, right? A set of connected elements that kind of bring together elements that were disconnected in the prior literature, right? So this is the background literature that's kind of drawn together by this particular piece or a study of rats that induces liver injury as a function of their being exposed to a particular chemical. And then we tried to kind of figure out, well, what is it that the system is doing? How does it move from the past hyper edges, so the past papers to the papers in the new space? Well, it could be that things are combining as a function of the common neighbors in this hypergraph. But it turns out that if we actually simulate a random walk across this entire space, rather than just looking at the number of common neighbors in this graph, that we're able to predict with logistic regressions about 95% of the new elements that end up combining in new papers, right? So we're using this hypergraph to kind of say, oh, how does the past research availability, right? So if researchers were randomly walking on this space, what's the likelihood that they would kind of come up with the papers they end up coming up with? And we're able to predict about 95% of that in terms of the area under the curve, in terms of the predictive accuracy at the later period. Much better than we were able to predict by something like common neighbors. What this suggests is basically, if people in the research are not attending, basically, if they're not attending to the needs of society, what they are attending to is prior research, Right? Prior research really constitutes the kind of the neural network on which science thinks as it moves from past research to new research. So another thing you can do with these kind of combinations of elements, in this case, we take just the chemical elements, right, and we look at the combination of those things across papers in time to get at a finer sense of what the algorithm that kind of describes what it is that authors are doing. So here's a chemist, and here are a whole family of chemicals that are combined in various papers in various ways, right? So sometimes, it's the discovery of zinc fingers here, the kind of connection of zinc and RNA. It could be therapeutics, so much less frequent chemicals, in this case, bromodeoxyuridine, with something that's highly central, like DNA, et cetera. So you can see a host of different kinds of structural traces that come as a function of papers drawing together these various chemicals. And this shows that these combinations end up being deeply embedded in other structures like time and space, right? So for example, these combinations which take place in Germany end up kind of linking over to the UK, that kind of bleeds into Canada, that kind of eventually combines in this particular 1996 paper these two these two drugs in broadly similar family. So anyway, so one of the things that we do or can do in this space is kind of look at the distribution of strategies that scientists perform. So in this example, you can imagine them combining chemicals in a variety of different ways, combining chemicals that have never been combined before, repeating former combinations, bridging subfields, or consolidating knowledge within a subfield. And what we find is when we look at these strategies across this whole space of PubMed documents, that we find a very stable distribution of strategies over time, which is surprising given the fact that there's an explosion of new chemicals that ends up entering this study, an explosion of new opportunities to link subfields and engage in kind of novel combination, which suggests that these communities end up focusing increasingly on repetitions over time. We find also that there's a correlation between the kind of the risk of a strategy, right? The unlikelihood that it's going to yield a publishable paper. So you're jumping to a new chemical. You can imagine going through hundreds of these kinds of experiments before achieving something that's publishable. Whereas if you do a repeat and extend and develop into something, it's much more likely to kind of yield a publishable insight. And so you end up seeing the risk being associated with the likely citations. However, those citations don't account for the full amount of risk that takes place in the scientific space. What looks like it does a much better job of accounting for the risk that actually takes place is if you map these strategies onto the full complement of high impact publications and prizes. So we link this to all the international prizes in biology, chemistry, and medicine over the last 50 years and find that elite prizes like the Nobel Prizes are much more likely to kind of introduce brand new chemicals or newly consolidate elements within a particular subfield than other classes of papers, right? So this is the average distribution of those papers over time. You can also use this distribution to cut through and look at how it is that how one might think about the efficiency of discovering the full space of combinations over time. So here we just look at the relative importance or centrality of chemicals and their distance within this graph of science. And we build a simple probabilistic model that I'm not going to describe here, but we use it to kind of cut through this variation to see if we can look at alternatives, right? We use that model to not only estimate the way in which science explores this space, but also to search through the space of alternatives. So we take a subset of the graph that's kind of matched on its distribution of degree and distance, and we use it to kind of estimate the actual strategy that we see in Medline. We also did it for US patents, which is a very concentrated strategy where people focus on central things. And then we search that parameter space to identify other more optimal search approaches. And we're able to find much, much more efficient search approaches, which is to say approaches which require to perform far fewer experiments to come up with the same relative proportion of the total graph of claims that we find in the literature. And in fact, one thing that you can see also is that the actual distribution ends up going in precisely the opposite direction historically that the optimal distribution goes, right? So for example, our probabilistic model suggested that things should, if you were optimally searching the space, you would actually look for less central things over time and you would take longer walks in the space of that network. And historically, you see precisely the opposite, right? As you estimate these from actual data, the scientific community is looking more and more closely at the center and most important chemicals, and they're having stronger and stronger anti-preferences for walking more distantly in this graph. I'm going to jump to the quick. There's also things you can do in the context of concept normalization, synonymy, which kind of reveals things about the game of science. So for example, when we try to parse phrases into one another, we discovered that there was kind of a, that there's an entropy, right? So there's some phrases that parse very cleanly into one another in some fields. In other fields, they parse very poorly into one another. And in some sense, the reason we're starting with biomedicine is because biology, medicine, and chemistry are the place in which words end up parsing most cleanly into one another, which is to say that what you mean when you use any particular word ends up being much more templated, which is to say its synonyms are much more precise than other fields like the humanities and the social sciences. I'm just going to kind of close with how it is that we might think about using all this information to kind of weight and search over the space of empirical claims, and in this case, in the search of claims about cancer. And so I'm just going to, this is, we're at the space of kind of research design and gathering data. This isn't the space of findings, but I'm just going to kind of lay out our search approach and then turn it over to Shanu, who's going to talk about, given kind of these weighted claims, how do you think about turning them into experimental hypotheses? So how do you, so it may be that some claims, as a function of things that we've talked about, are kind of like, they're robust, and other claims are less robust. And so there's different ways in which you can imagine these claims being altered for later, to generate experiments, because people say that they're uncertain about those claims, because maybe there's greater independence in the contribution that leads to those claims, because investigators or methods may be more or less accurate in their kind of track record of historical claims, and as a function of the selection of findings, right? So the weight of these claims ends up being a complex function of these different things. So independence, you can imagine that things can be technically independent if multiple methods end up being used to find the same claim, to support the same claim. Socially independent if different authors, networks, and kind of lineages within the scientific space independently corroborate the claim. Or culturally independent if different journals, different communities, different literature ends up being garnered to support the research design that ends up supporting the claim, right? And so then you can kind of discount the claim in terms of its frequency and volume accordingly, right? So if it's much less independent, if it's much less socially independent, if it's basically a single group that's recruited other people over time, if it's basically the same set of conditioning experiments and citations, then you would expect it to be less robust over ones that represent a much stronger support. And we find in previous literature, which I'm not going to describe right now, that this appears to be the case. So this is a role of beta amyloid in development of Alzheimer's plaques, which ends up looking as a function of its lack of independence, its lack of a role in this process ended up being overlooked really over time. You can also think about the accuracy of authors in general or on specific tasks, of teams in general, and institutions in general. And we actually use a variety of probabilistic models that Andre and colleagues developed, and that together we've kind of developed some software to induce, as a function of agreement over time, how to estimate the relative firmness of an actor. And then finally, selection of claims, right? So what findings do journals select? Well, they select findings often as a function, and this is a cartoon really, but of surprise and support, right? So if you get an article into Nature, if it doesn't have much surprise, it better have enormous support. If it doesn't have enormous support, it better have remarkable quantity of surprise. So it's a product of these two things. And insofar as we're able to measure ex-ante novelty as this kind of inverse, right, of the probability of this discovery, then let's see, I'm not going to talk about all those elements. Well, okay, yeah, so insofar as you're able to estimate that, then you can back out the likely support that was required for that article to be placed in that venue. So with that, I'm just going to kind of turn the time over to Shauna. So given that you've got a set of claims now that are kind of robust, you understand the system in which they live, then what do you do with them? How do you use them to make predictions about new treatments in this case?